Good morning, Droitwich, again. As was just said, I've told you before about some aspects of my hobby. I've told you about telescopes. I gave you a talk about the Star of Bethlehem. This is the day job. So I work at the University of Liverpool. I retired a little while ago, which means I no longer do any undergraduate teaching, but I've kept all of the connections on the research side, and this is what I'm telling you about. Research that started quite a few decades ago, but is essentially ongoing. And as you can see, I'm talking about essentially imaging, essentially microscopy. So it's completely the other end from astronomy, where we use telescopes to look billions of light years away. Here I'm talking about using microscopes to look at the microscopic world rather than the macroscopic world. So in a sense, we're going from where I started, which is using microscopes for looking at atoms, building up to look at molecules, and then looking at biological systems. That's the way we're going here. So I'm going to start from where I started back in the late 80s, early 90s was where I started my research, first at the University of Birmingham, and then I moved to the University of Liverpool, where I've been for the last 35 years or so. I'm going to take a little aside to tell you why it is that analyzing images, regardless of whether they're being taken from microscopes or other instruments, why looking at images can be problematic, because what we think we see in an image is not necessarily what is there. So I'll do a little aside about perception of images. And then, although I started as a physicist looking at atoms and molecules, I soon found that when you're looking at images, there are often spin-offs into other disciplines, not necessarily physics. It could be chemistry, it could be engineering, it could be one of dozens of other disciplines where taking images is a key part of how the science progresses. Then I'll be looking at the work I've done taking imaging into the world of living matter, looking at effectively biophysics, biological physics, and I'll be finishing with some work that I'm doing now on investigating diagnosis and prognosis of cancer. So where should we start? Well, the talk at its heart is about how we deal with images, the scientific approach to images. Rather than looking at a photograph saying, isn't that nice? We're looking at images saying, what information can we extract from those images? And two concepts are important. I'll be talking along the way about image processing and image analysis, and it's important that we get that distinction sorted out. Image processing means if we take an image, sometimes that image doesn't convey a lot of information. We look at that image and it's perhaps difficult to see what that image is or to see the information we're trying to extract from the image. Why did we take the image in the first place? Image processing means taking an image and doing something to that image, usually for the purpose of making that image more clear, for making that image a little bit either crisper or a little increase in clarity, or perhaps simply changing the colors or the contrast in order that we can extract information more readily from that image. So image processing is take an image, do something to it, and produce a second image which hopefully is easier to interpret. Image analysis means taking an image and extracting something quantitative. In other words, I might take an image of a whole load of blood cells, and I might be interested in how many blood cells are there in this image. Maybe I want to actually just count them. In other words, an image is the starting point, but the output is not another image. The output is just a number. And in many cases, the scientific reason for taking an image is to extract information and convert that image into a set of numbers which is in some way useful for the person who took the image. So we've got image processing and image analysis. They often get conflated because it is quite common for people to do both. Perhaps you do some image processing in order to do some image analysis, in order to extract some information from those images. And this talk will be illustrated with a few images I've taken, a few images I've borrowed from other people, other collaborators who I'll acknowledge at the end of the talk. And I'll be showing some images from essentially the past 40 or so years. This happens to be not one of mine, but it's a very nice image which shows you, in effect, the state of the art. It is possible now to build microscopes in which you get magnifications as indicated here. Look at the scale of the size of the image. The image is less than 1 nanometer from end to end. That's 850 picometers. And pico, remember, is 10 to the minus 12. So the entire image is less than 1 nanometer being 1,000th of a micron, 1 micron being 1,000th of a millimeter. So in effect, the magnification, if you want to think of it in terms of that image as it stands there, the magnification is a little bit shy of times 1 billion. Okay, so you might be used to the idea of microscopes magnifying times 10, times 100, times 1,000, times 10,000. This is something like times a billion. And we're seeing individual atoms of, in this case, carbon. So these carbon atoms are obviously relatively small. They are only a few picometers in size, or a fraction of a nanometer in size. And we can see a dozen or so of them in this particular image. Carbon happens to be a material that's quite easy to image, so when people test their microscopes to see how well they work, they often look at carbon. But just again, to give you a sense of scale, if we took this particular image and imagined, well, at this particular magnification, how big would a grain of sand be? The answer is it's about the size of the moon. That gives you an idea of the sort of magnification we're dealing with here. And whenever I look at images in which we're looking at individual atoms on a surface of a material, I often hark back to the William Blake poem that starts with, "To see a world in a grain of sand," because at this magnification, a grain of sand is a huge number of atoms, a huge number of molecules. So that's a starting point which tells you just what magnification we can get. And microscopes like this, they're called scanning tunneling microscopes. I'll explain that in just a second. But these have been around for a few decades now. They were invented in the 1980s. So for the last 40, 45 years, we have been able to build microscopes that allow us, you can see what talks I normally give, we can build microscopes that allow us to resolve individual atoms. The process by which that happens, with normal light microscopes, we're limited to a certain resolution determined by the wavelength of light. But these microscopes are not limited to that because they work via a process which I am not going to describe in great detail, but it relies on quantum theory, it relies on quantum mechanical principles. So in a sense, we're using quantum mechanics to allow us to see quantum mechanical objects on a scale of atoms and molecules. There's the scanning tunneling microscope in the background, almost hidden by the handsome scientist standing in front of it there. This, as you can possibly tell, was taken a few years ago, back when I had a moustache and less of a beer gut, but there we go. But the scanning tunneling microscope, again, I'm not going to go into a huge amount of detail, but you can't see it because it's inside a vacuum chamber. Because we are looking at individual atoms, we do not want all of the molecules in the air getting in the way of our imaging this particular material, whatever it happens to be. And so all of the action goes on inside a vacuum chamber. And the vacuum inside these instruments typically is 10 to the minus 13 atmospheres. In other words, it's less than 1 part in a trillion of the pressure of air we're dealing with here. If you remove most of that air, then you're left with a good enough vacuum in which you can do these sorts of experiments. How does it actually work? Well, again, I'm not going to go into too much of details because this is not a physics lecture per se, but what happens in a so-called scanning tunneling microscope is a microscope is self-explanatory. Scanning means something moves backwards and forwards over the sample to image it. It's not a snapshot. Something is moving backwards and forwards. So these are called scanning microscopes. And in this particular case, a very sharp tip is brought close to a surface. So the red little pyramid at the top there is a very sharp tip of material which is brought close to a surface, and that's the surface we are trying to image. The surface, generally speaking, is not flat. It might have mountains and valleys in it. At the very least, it is made of individual atoms, and each atom is essentially a bump. And although these, of course, are very small, you can see that if we were to move a sharp tip over a surface, the distances will change. If we're moving over a mountain or over a valley or over a mountain or over a valley, the gap there between one and the other will change. And a tunneling microscope works because the word tunneling is a quantum mechanical expression, which means that although in principle electrons will not flow from one to the other, if we try and make a circuit and connect the tip to the sample, there's a gap between the two, so it's open circuit, so no current will flow, according to Isaac Newton. But according to quantum theory, a small current will flow. It doesn't matter that it's a small current. What matters is it is very sensitive to how much they are separated, that D value. You can change that D value by a fraction of an atom, and you can measure how much that changes the current that flows if you were to connect the tip to the sample via a very sensitive ammeter. The process by which that sensitivity comes about is purely a quantum mechanical effect, and unless you've got a few hundred hours, I haven't got time to explain all of what's going on. But the bottom line is, by measuring the current as a tip moves backwards and forwards across a sample, we can actually measure very, very small details. For instance, here is a small slab of material. The scale there, if you can't read it, is 2 nanometers. So again, we're only scanning a few nanometers. We're not scanning millimeters of sample. We're scanning tiny areas, but in the process, we see little mountain ranges on the surface. This particular surface has been colored artificially, and it's colored this goldy color because this is the surface of rust. Rust is an iron oxide. This particular surface is a combination of iron oxides. It's Fe3O4 and Fe2O3 for those who like their chemistry. And each little mountain, each little pyramid that you can see that appear to occur in islands on the surface, each of those pyramids is a single atom. So if we wish, we can scan a surface and we can see where the atoms are. This is an enormously powerful tool. And before the 1980s, we had to guess or infer where the atoms are in the surface of a material. Now we can actually see where they are by doing scans like this with microscopes like this. So I must credit the fact that, again, this wasn't one of my images. This was from a colleague from the University of Manchester. But along the way, I started using scanning tunneling microscopes back in the early 90s. And I realized that I wanted my PhD students to be able to analyze these images that were coming out of the scanning tunneling microscope, hereafter simply referred to as an STM. So I figured that we needed some software that allowed our students to be able to look at the images and play around with the images and process the images and analyze the images. And there are lots of different types of scanning microscope. There's a scanning Auger microscope. Auger is a word that refers to a particular type of electron which is kicked out when you shine X-rays onto a material. And perhaps you've heard of scanning electron microscopes. They are the more popular type of electron microscope. These have been around for a long time. Even before scanning tunneling microscopes came along, the SEMs have been around for decades earlier. And there are other types of microscopes, scanning force microscope, where again, you move something over a surface, and in this case, you measure what's the attractive force between a tip and a sample. And amazingly enough, they can also give you fantastic resolution. And there's loads of other scanning microscopes, scanning ion microscopes, scanning laser microscopes, scanning optical microscopes, scanning tunneling microscopes we've always mentioned. In fact, there's a whole raft of scanning something microscope. If you give me a letter of the alphabet, I can probably put something in the middle there and tell you there's a microscope that fits that bill. So there are dozens of different types. There are a big family of scanning microscopes which all work on slightly different principles. But I wanted my students to be able to look at images from any of these microscopes. Microscopes they might have used themselves, and possibly images from some of their colleagues using other microscopes. So I figured that scanning something microscopy was very important, and that led to this acronym. Instead of calling it STM for scanning tunneling microscopes and SEM for scanning electron microscope, I thought, let's just bunch them all together and call them SXM. And that led to a piece of software that I wrote back in the early 1990s, which was this unpronounceable image SXM. There's no way of pronouncing SXM, and the X simply means any of these types of microscopy. I made it available because although I wrote it for my students, I figured that once my students had used it for a little while and got all the bugs out of it, I figured, well, this actually might be useful for other people in the world as well. So I made it available, I stuck it on the internet, and then watched people download it. And I realized that people were interested in this software. Because of this burgeoning STM coming around in the 1980s, there were lots of labs around the world that wanted to be able to look at their images. And these are the countries in which I know people are using my software. So obviously a lot in Europe, a few around the world. I'm having difficulty getting into Antarctica, you see. There's nobody there that seems to want to use it. But thousands of users in quite a few hundred different countries around the world. So it's very satisfying. This is over a time period of a few decades. It looks like people definitely use this image processing, image analysis software. What I spent a lot of my time doing in the early 1990s was looking at a batch of metals called rare earth metals. I started this research when I was in the University of Birmingham. The rare earth metals, again, we're not going to go into details. These are the ones at the bottom of the periodic table that everybody sort of forgets about. I figured there was a little bit of a niche here to start looking at these metals because other people were concentrating on other metals in the periodic table. I started looking at the rare earths and I started trying to analyze where are the atoms arranged on these various metals that exist in the periodic table. And it's not just stamp collecting in terms of, okay, the atoms are arranged like this or they're arranged like that, because ultimately the positions, the arrangements of atoms on a surface are intimately linked to the properties of those metals. If you know where the atoms are, in principle, you can calculate the chemical properties, the mechanical properties, the reactive properties of that particular surface, or the magnetic properties. If you know where the atoms are, you can start understanding the various properties of the various materials. And that's one of the reasons I was doing this, to try and get a better understanding of how these particular metals behave. I did some work with scanning tunneling microscopy. I used other techniques as well. Scanning tunneling microscopy allowed me to look at the surface, and I found that the surfaces were not quite as clean as I was hoping. These little blobs here, these white blobs on the surface, are little bits of oxygen that are contaminating the surface. The rare earth metals are very reactive, and even under the best vacuum conditions, it's still almost impossible to get them completely clean of any contamination. So I was using microscopy. I was also using other techniques as well. I was using spectroscopy, looking at how the distribution of energies of electrons in these metals determine their properties. I was also using diffraction techniques, which is a way of getting some information about how the atoms are arranged within the surfaces. In many cases, in many situations in science, it is not uncommon to use multiple techniques which all give you slightly different information about a system, and then you pool all of that information to try and get a better understanding of what this system is. To give you an idea, one of my colleagues, when I was working in the early 90s, was working on alloy systems. I was interested in pure metals, but my colleague was interested in alloys, and he took some scanning tunneling microscope images of surfaces of an alloy of aluminium and palladium and manganese, and he was trying to figure out where are the atoms in this particular system. And in this case, you can hardly tell. Again, white blobs mean contamination. That's almost universal. But the question is, are these particular smudges in the background, which are the individual atoms of this particular alloy, are they arranged in any particular pattern? And with all credit to my colleague, he produced much better images than this, but I love this image because it's not good. And then you can ask yourself, how can we process that image? Are there any mathematical tricks we can apply that will allow us to get better information about where the atoms actually are? Because it's very difficult to tell as it stands at the moment. And again, without going into the maths, I know this is a maths group, but I'm sure you don't want pages and pages and pages of mathematics, but it is possible to process images and effectively ask the question, if we expect the atoms to be arranged in particular patterns, and in this case we had reason to believe that they are arranged in patterns of 5, like pentagons, or patterns of 10, like decagons, if we think that's the case, can we find out what those particular patterns are, those so-called symmetries of the situation we're dealing with? And it is mathematically possible to take an image and effectively extract what are the particular frequencies in terms of how things are arranged in space, and we can get a so-called diffraction pattern, and then we can use that to say, well, it looks like these dots in this particular diffraction pattern are telling us that there's a particular tenfold symmetry. There are 10 dots in each of these rings, implying that the entire image can be rotated by 1/10 of a revolution, or 2/10, or 3/10, and a lot of these atoms will produce patterns that fall back onto themselves. And the wonderful thing about mathematics is as soon as you start extracting information like this, you can start to say, well, if there is something in there that's tenfold symmetric, can you tell me which particular atoms seem to be obeying this tenfold symmetry? Because the blobs of oxygen are just positioned at random. So can we extract the information and see what that tenfold information is telling us? And that's the same image transformed back again, but this time preserving only the bits of the image that have this tenfold symmetry and throwing away everything else, which means all of those oxygen blobs there that are positioned at random have disappeared because they are not tenfold symmetric. And you look at this and you say, well, now it's a lot easier to see where the atoms are by looking here compared to looking there. But we do have to be careful. Are we sure we're not kidding ourselves? We are manipulating the image. Well, we know about image manipulation for the last few days, don't we? People don't like images being manipulated. So are we sure that we're not simply imposing a pattern? Are we sure that we're actually bringing out what was already there in the image, or are we just imposing some new pattern on top? And if we look at the orange, there's no reason it's orange other than it looks pretty. If we look at that and we look at various parts of it, we can distinctly see some parts which are pentagons, and the one circled there is a decagon. Can everybody see a 10-petaled flower in there effectively? So for some reason or another, the atoms want to arrange themselves in rings of 10, if we believe that is correct. And we can ask ourselves, are we actually enhancing detail that was already there, or are we just making up tenfold patterns by doing our mathematical analysis? Well, if we look with exactly the same pixel position at the original image, what do we see? Well, it's a rather ropey version, but I hope you can still see that we've got a tenfold daisy there, or sunflower. It is actually there. We are simply enhancing the detail, making it much more easy to see. We are not imposing anything new, but we do have to be careful. Whenever you do any image processing, you always have to ask yourself, are we over-processing? Are we actually imposing things that weren't actually there in the first place? So whenever we do analysis like that, it's perfectly okay to publish that image and say, look, these are where the atoms are, but we do have to be careful and we do have to do checks to make sure that we're not kidding ourselves. I also did some work with colleagues who were interested in how molecules are sitting on surface, because that's ultimately important to how molecules react with each other, reactivity of a surface, catalysis, things like that, depends on how molecules are sitting. And this particular individual was interested in a molecule which can sit on the surface in 4 different ways, 4 different patterns, if you like. It can be this way up or that way up, and it can be left-handed or right-handed. And they were interested in all of the molecules we can see in the image. Are they any particular subset of those 4? Is it left-handed is more likely than right-handed? Is there any so-called chirality? So I analyzed this image using image SXM, and by putting the symbolic triangle on the left-hand side, we then looked at the result, and the answer is, not really, no. There's as much red as there is blue, and there's as many triangles pointing up as there are triangles pointing down. So in this particular case, we have analyzed the image and come to the conclusion, no, there is no particular handedness, there is no particular preferential orientation of this molecule. If there was, that might have led to some other avenue of research, but this is the sort of image processing and image analysis that I did a lot of in the early 1990s, processing my own images and sometimes images of colleagues to try and extract information of interest. But why is it such a big deal? Can't you just look at an image and figure out what's going on? And the answer is no, because unfortunately perception and reality are quite often different. And there are plenty of circumstances where we find what we think we're looking at in an image is not actually what's there in the image, because our eyes and our brain are doing a huge amount of image processing that we are not even aware of. It's just what your brain does to keep you alive and to stop you getting eaten by lions or stop you tripping up. There's all sorts of processing being done, and it's being done as a process for the last 500,000 years. Our brains have developed to do that to try and increase our survival prospects. So you can ask, well, which is better then? Should we be analysing images with our brains by looking at them and figuring out what's going on? Or should we be using a computer, in which case we've got to be careful that the computer is giving us the answer we expect? And you could say, which is better? Well, better is a very subjective thing. They're different. And it's a bit like saying, well, they're both computers. The brain is one computer that's made of carbon, and that particular computer sitting on the table in front of us is a computer made with silicon. So is it better to use carbon or silicon for processing images? And the answer is they give very different results. So with apologies for those that have seen this before, let me illustrate the fact that what you think you see is not necessarily what's actually there. There's a couple of sort of Rubik Cube type things there. Can I just check, is anybody colorblind? Sorry, sir, this is not going to work for you. I don't think it'll work for you. You can tell me afterwards if it does. We've got a couple of Rubik's Cubes with various colored tiles on them. On the left-hand side, I'm interested in the colors on the top face. To hell with the other two faces. They don't really mean anything. So we've got a load of colors on the top face of this cube. How many of them are blue? How many of those little tiles are blue on the left-hand image? Apart from 1, 3, I think we've got a 4. You can see 4 as well, sir. Okay, that's a good start. So we've got 4 blue tiles on the left. On the right-hand image, again, we've got a cube. And again, I'm only interested in the top face of this cube. How many of those tiles are yellow? Okay, 1 or 2 6s, and then we get 7. Interesting. A few more for the gentleman at the back. But okay, most people would say 4 blue on the left and 7 yellow on the right. Okay? Fine. Are you prepared to put money on that? You may be interested to know that the 4 blue tiles on the left and the 7 yellow tiles on the right are actually the same color. And I mean that genuinely. They are genuinely the same color. All 11 tiles that you think are the 4 blue ones on the left and the 7 yellow ones on the right are actually the same color. And you're not necessarily going to be convinced by me, but you can go to this web page and check it out for yourself. What I'm going to do is take away all the pixels other than the pixels in the 4 blue and the 7 yellow. And if I take all those other pixels away, that is actually what you were looking at. You were looking at 11 gray tiles. And you only thought they were blue, or you thought they were yellow, because of the other pixels that are throwing you off. The other pixels in the background, clearly there's a lot of yellow pixels on the left, there's a lot of blue pixels on the right. And it's purely the contrast. Your brain is telling you, well, they may not be blue, but they're certainly bluer than all of the other pixels in the neighborhood. And on the right-hand side, well, they may not be yellow, but they're certainly yellower than all of the other pixels around. Your brain finds it impossible to actually disentangle this, because your brain is built to give you the information it thinks you want to know. And your eyes and your brain do not tell you, when I'm looking at this thing over here, there's this much red and this much green and this much blue. It's always making comparisons between different colors. And therefore, you cannot believe your eyes. I told this to a postdoc who had spent the last 3 or 4 months actually looking down a microscope, picking out cells that were blue and cells that were gray. And then I showed him this, and then he went sort of pale. I'm not sure what color, pale, but he went pale, and realized that everything he'd been doing for the last 3 months was useless because it was subjective, not objective. And then he realized that he really needs to think about using a computer to do the analysis, which can actually tell you what that color is, regardless of what the neighboring colors are. And for those that think, well, okay, that's the situation with color, but at least I know the difference between bright things and dark things, regardless of whether I get the color right. Well, no, you can't. Let's just go back. That's just a reminder of what those 2 sets of tiles were. But let's look at an example in which we're not dealing with color, but just dealing with shades of gray. Perhaps, again, you've seen this. It's a very common one. If we're looking at the square labeled A and the square labeled B, we're not interested in the letters. It's the squares on which those letters are sitting. Is the square labelled A darker or brighter than the square labelled B? Aha, I see people laughing now, yes. You can sort of see what's coming. Let me, again, without touching the image itself, let me just bring in a couple of masks on the left and another mask on the right. Now that I've done that, what do you think the brightness of square A is compared to the brightness of square B? Each of them is identical to the slab on the left and the slab on the right. They are actually the same grey. They are not similar, they are absolutely the same shade of grey. If you go into software and check, the pixel values are identical. So now that you know that square A and square B are exactly the same shade of grey, let me take those two masks away. Okay? Now you absolutely know that A and B are the same shade of grey. What is your brain telling you? Even though you know the answer, it is impossible to override your brain telling you, well, it looks like the B square is actually brighter than the A square. That is why it's so difficult to do any image processing and image analysis, in the sense that what you think is in the image is probably not what's actually there. And this came across when somebody asked me to do some analysis for them. They said, I'm interested in these little squiggles, the DNA strands. They had strands of DNA deposited on glass slides. Now, you think glass as being sort of polished and absolutely flat. Well, it is on a scale of a millimeter, absolutely flat. But if you're doing something in which you're looking at nanometers or a fraction of a micron, basically glass is rough. It's all a question of scale. So these mottled grey areas in the background are simply the background glass. But on top of that, you can see these white squiggles. I assume everybody can see those okay? If I gave you long enough, I'm not going to do this, but if I gave you a minute or so and asked you to count the squiggles, you could probably all do that. You could look at it and you could go, and you could probably come to the conclusion, yes, there's about 30, 40, 35, 40, somewhere around there, about the same as the number of people in this room, very roughly. And nobody would have a real problem saying, yes, I can see, let's say, 40-ish squiggles. And the assumption was that, well, you can get a computer to do this. Why should I sit at the microscope and write down the 40 squiggles on that image, next image, 50 image, okay, etc., and spend the next 6 months of my life doing that? Surely a computer can do that very rapidly. And the answer is, well, a computer can do that, but it's nowhere near as easy as you assume. Because when you look at that, you say, well, all of the DNA is brighter than the slide, so basically all you need to do is to look at how high something is or how bright something is. For an image like this, height is the same as brightness because of the way it's been presented. Bright means high, dark means low. So you look at that and you think, well, if I were to take a slice, if I were just imagine a profile through one of these lines, what I'd be looking at is something that looks like that. There would be background glass slide, which is a little bit mottled, and then every so often I would come to a DNA strand, and then there's a bit of background, and another DNA strand, and then a bit of background, and another DNA strand. So surely all I need to do is to get the computer to say, pick out all of the bright bits, pick out all of the peaks here, and then I can just count them. Surely that's going to be easy. Well, it looks it when you look at it, but what you're actually looking at is not a profile that looks like that. What you're actually looking at is something more like that. And you're thinking, which of those spikes is a spike because of DNA, and which of them are from the background? Surely that can't be right, because the background is grey and the DNA is white. Well, no, it isn't. It only looks like that because that's the way your brain is interpreting the image. This particular profile here, this is over 1 micron. So this image is a few microns. Over 1 micron, I've looked at the height, which is the same as the brightness, and the particular profile I've taken is that one. And that profile is a lot of nothing, and then there's a DNA and a DNA and a DNA. So there's actually 3 DNAs in there. I can point them out with an arrow. There's 1, there's 2, there's 3. Which of those spikes in the profile corresponds to those 3 DNA strands? The answer is that one, and that one. How on earth is the computer supposed to figure that out? you can work it out in an instant by glancing at that and saying, well, there's a strand, there's nothing, there's nothing, there's a strand, there's a strand. But the computer would find it very difficult to figure out that that particular peak there corresponds to a DNA, but the one next to it, which is a bit higher, isn't. So, okay, the highest one is indeed DNA, but what about all the others? So this is one example of what you think is going to be trivial actually turns out to be really complex. Sometimes you get it the other way around. Sometimes you find something that looks like it's going to be really tricky to do. The computer basically just has it for breakfast and can do it very easily. But quite often you find this situation where the human judges that it should be an easy problem, but the computer finds it very difficult. What I had to do here was to do some processing in order to make it easier to see what was actually going on. What I actually did was to say, well, what is the characteristic of one of these strands that separates them from the background? And I figured that one of the characteristics is the fact that these strands are effectively little cylinders. They may be wiggly, but they're effectively cylinders. And if you think about the height above a background, if you were to move along the DNA strand, the height remains the same. And if you go sideways, whoa, the height suddenly changes. There's a very dramatic change of height if you go one way, but a relatively slow change in height if you go the other way. Now, you have to work in the fact that these are all wiggly rather than straight, so it's not quite as simple as that. But that's the process I used to figure out how can I tell if this particular pixel here is part of a rough background or is part of DNA. And using that idea of does it change its height dramatically if I go this way compared to going that way, I then figured out this pixel is probably DNA, this pixel is probably not DNA. And once I'd done that, it was possible to produce a map of where I think the DNA is. As soon as you've done that, it becomes genuinely trivial for the computer to count them. Because, of course, not only can we pick them out more easily like that, but the computer, all it has to do is to count the black pixels, and it's effectively got the total area. Or it just works out, if that's a bit of DNA, I just trundle along here until the black pixels stop, and that's how long my DNA strand is. So, as long as you've done the image processing, which is the really, really tricky bit, once you've done the image processing, then doing the analysis, counting the strands and working out how long the strands are or how wide the strands are, that's relatively straightforward. But one nice example of something that you think is going to take 10 seconds, it took me weeks to work out how to do that. So let's have a look at spin-offs. I've got here spin-offs in earth sciences and medical sciences. I'm not going to cover the earth sciences today. I've got a sort of separate talk which concentrates more on the earth sciences. But as I was developing the software, I realized that not only was it useful in physics and chemistry, but it also, in principle, had applications way beyond that. Everything from anthropology up to zoology. Again, I could virtually fill the alphabet with all the disciplines that could benefit from this particular approach. So I started working with other people on applying it to earth sciences. And I started working with medics within the University of Liverpool to start thinking about how the image software that I wrote could actually be applied in various medical applications, which ended up under these particular acronyms. I particularly like this one, the microscopy image analysis software for medical applications, which gives me miasma, which is wonderful. The medics hate it, but I thought it was a wonderful acronym. So I'm not going to be talking about earth science. I collaborated with a colleague in Switzerland. We ended up writing a book on image analysis, available from the author at very reasonable cost. But today I'm talking more about the medical spin-offs. And over the years, mainly in the this is not so much in the 1990s. Most of my work in the 1990s was working on rare earth metals, but towards the noughties, after the turn of the millennium, I started working on medical applications and found a lot of different projects in which people wanted to know the answer but didn't know how to analyze their images. So I worked on particulate matter. This is trying to work out how much carbon there is in your lungs if you inhale smoke, not smoke from smoking, but smoke from burning fires, for instance, in Africa, where they cook all their meals over an open fire, how much carbon ends up in their lungs, trying to quantify that. I also worked on malaria parasites, trying to automatically count malaria parasites rather than having lots of PhD students staring down microscopes with a clicker saying 1, 2, 3, 4, 5, 6. And yes, they did do that before the software came along. Microcirculation is something I'll say in a little more detail in just a moment. I also did some work on retinal analysis because of course, there are lots of images of retinas these days, especially now that every optician gives you the option of taking a retinal scan. So there's lots of images of retina, and if you look at retinal images and look at the blood vessels and look at irregularities on the back of the eye, they can sometimes be a precursor for other diseases like diabetes, for instance. So there's good reasons for doing retinal image analysis. And even though I didn't spend much time myself doing that, I did have a number of project students working in this area for a few years. I also looked at the morphology of parasites. The earlier one was on malaria parasites, but I also worked on the morphology of Leishmania parasites because the parasite changes its shape and size and has a tail that changes length over its life inside the host, and so by analysing what the shape of this parasite is, you can get an idea of how long the host has been infected. I also worked on various other areas, but one I just particularly want to pause on is one of the later ones I did, which was looking at human chromosomes. This is a false colour image. They're normally just imaged in grayscale. I've just coloured it to make it a little easier to see what's going on. I worked with Liverpool Women's Hospital, who said, we spend our time looking at images like this. They regularly scan pregnant women to try and find out if there's any chromosomal abnormalities in the unborn child, in the unborn fetus. And so experts who've spent years doing this literally spend their time looking down a microscope and looking at that particular collection of 23 pairs of human chromosomes, 46 in total, saying, does that look right? And they, amazingly enough, can tell at a glance, almost at a glance, it takes them a little while, but within half an hour, they can look at that and other images similar to that and say, all of this looks fine, except it looks like there's a defect on chromosome 9. And it's amazing that people can actually do this. But this is a reminder of how good humans are at pattern recognition. What they're actually doing is comparing it with either their experience or the fact that they know what a standard human chromosome should look like. In other words, all chromosomes in the human genome are mapped out according to their length. Chromosome 1 is the longest, 2 is the next longest, 3 is the next longest, etc., all the way up to 22, and then you have the sex chromosomes XX or XY. And they were saying, we spend a lot of our time, virtually 9 to 5, staring down a microscope, looking at these, trying to figure out, well, that's chromosome 1, that's chromosome 2, that's chromosome 3. Where the hell's chromosome 4 gone? And where's its partner? Remember, there's 2 of everything except for the sex chromosomes. So there should be 2 1s, 2 2s, 2 3s, 2 4s, etc. And basically, they look at that and they map them all out. And they said, wouldn't it be nice if we had some automatic way of doing this? And I was still rather naive at this time and thought, yeah, that should be possible. But then I'd forgotten this adage that what looks really straightforward, not necessarily quick, but what looks straightforward, all you have to do is to figure out the banding pattern, which is coloured here, and match it up to the banding patterns here. And if anything doesn't match, then you've got a chromosomal abnormality. And I thought, well, that might take a little while, but it's worth having a project student on it, and I'll dabble with it myself. But the one thing I had sort of overlooked was just how problematic it can be when you end up with a set of chromosomes, and this is about as separated as they get before a particular cell splits during mitosis. The problem is, as you can see in a number of different cases, some of the chromosomes overlap each other, which means you're trying to work out what a particular pattern is in terms of light, dark, light, dark, light, dark on a particular chromosome. The fact that it's not straight doesn't help, but it is possible to take all of these wiggles and straighten them out. And you're trying to work out the pattern of light, dark, light, dark, light, dark. But some of these chromosomes are on top of each other, which of course means you're missing a little bit of information, because when one chromosome overlaps with another, you can see the banding pattern of the one on top, but there's no way you can get information about the banding pattern of the one below. So the problem I found was a lack of information. You can take this image, if they were all absolutely perfectly well separated, then in principle you could straighten them all out and then try and pattern match them to each of the chromosomes and then find out if any are abnormal. There are lots of these images. In other words, the particular people who take these images take lots of images of lots of cells in which the chromosome set has been separated out, but there's no relationship between them. If chromosome 13 is sitting over here in one image, there's no way of telling where it is in another image. So you can't simply say, well, I've got most of that pattern, but not that bit that's missing. You can't necessarily find that bit that's missing in another image. And this is the problem I found. It was rather frustrating that I could do a certain amount of work on pattern matching, but I always had this problem. If you've got a finite number of images and there's lots of chromosomes that cross each other, there will always be a problem of trying to work out what is in the missing section. And you notice that some of these chromosomes are very similar in length. You can't rely on the fact that that one is that long, so it must be chromosome 15, not chromosome 16, because they only differ by, as it were, a pixel or two. And it's not necessarily reliable enough to say, I'll just use the lengths rather than the banding patterns to work out what is what. So kudos to the people who spend all of their time looking down microscopes. They can analyze images like that in less than half an hour and tell you everything is fine, this pregnancy can go ahead, or this one has got an abnormality, this child is going to be born with a particular genetic defect. So I tried to work with Women's Hospital at Liverpool. It was interesting, but it never actually came to a final piece of software that I could say, this is what you need to take you away from the drudgery of doing all of this work manually. Let's have a look at some of these applications in a little more detail. I'm going to look at two, so as not to be here all morning. And the first one is microcirculation analysis, and the second one is cancer. When talking about what miasma could do in terms of I can write software and it can be applied to various medical situations, I got talking to a consultant at Alderhay, the Children's Hospital, and he said, we have a lot of problem, especially with youngsters who have sepsis, who have blood problems, what you might call blood poisoning, but basically just sepsis. Patients that suffer from sepsis have poor circulation. In other words, the blood does not circulate properly, especially noticeable in the capillaries, not necessarily coming from the heart, but in terms of capillaries feeding oxygen to the various tissues of the body, there is a problem. And the way that can be monitored, and the way it's standardly monitored for sepsis, is so-called sublingual microscope. And I thought, what is a sublingual microscope? Is that a microscope that doesn't know how to talk? Or, okay, sublingual means under the tongue. So under the tongue, the capillaries are visible. In other words, capillaries are all over your body, but they're underneath fat or skin or other things. Underneath the tongue is one place where the capillary network is most easily visible. And so a standard instrument that's been used for quite a few years is a microscope that's placed under the tongue to take a small section of the capillary network. And by small, I mean a millimeter or so. It is a microscope that's looking at a millimeter square of capillary network. And this is being shown to be representative of what's going on in the body. In other words, if you have a circulation problem and blood is not getting to tissues around the body, whether it be your arms and legs or other organs, it will show up by looking at what's going on under the tongue. So we're not really putting people under the microscope, we're really putting the microscope under people in this particular example. So there it is blown up a little bit, and a reminder that it's 1 millimeter or so. And you can see blood is flowing. In some cases, you can see quite happily blood cells moving. In some cases, they're moving so fast they're a blur. In other cases, you can see them moving through capillaries. But notice every once in a while, there are some capillaries where the blood cells do not appear to be moving at all. The problem with this particular microscope is, again, you look at this and you think, well, this ought to be straightforward. An expert can look at this and say, well, yeah, those seem to be flowing okay, and those seem to be flowing, well, a bit slowly, but probably okay. And this one appears to be stalled. Let's just rerun the video. They seem to be moving, okay, they're sort of moving here. Some of these things, the blood flow seems to be flowing backwards. Here it seems to be going forwards, backwards, forwards, backwards, forwards, backwards, not really doing much. Other areas seem to be okay. A person can look at this, and in a few minutes, an expert can look at that and say, this patient is getting better or this patient is getting worse. We're not trying to work out whether the patient has sepsis or not. This patient already has sepsis, because that's why this instrument is being used. What we're trying to do is to treat the patient and ask, is the patient getting better? Are the drugs we're using making it better or worse? Is the microcirculation, the circulation in the capillary network of this patient, is it improving or is it getting worse hour by hour by hour? With sepsis, if you didn't already know, it is crucial to get in quickly. If you wait 24 hours before changing the drug regimen, you could lose either a limb or possibly the life of the individual. So the question is, can you automate the process of looking at what these blood cells are doing? And when you first look at it, you say naively, well, yes, a computer ought to be able to work that out. But there's a lot of catches here. This particular microscope is set up to look for, effectively, hemoglobin. It's looking for blood cells. And so we're seeing blood cells, and that's why you see individual cells that don't seem to be inside a vessel of any kind. They seem to be just floating in the middle of nowhere. The vessels themselves are invisible. So you cannot actually see in this image any blood vessels. You're only looking at blood cells. If the blood vessel is full of cells, you see something. If the blood vessel has got a few cells in it, you see a few dots. But it's problematic if you were to zoom in to any particular part of this video and say, well, let's just see what it is we're actually looking at. Let's blow it up a little bit. Okay, I can sort of see something there. But if we say, well, okay, let's blow up a little bit more and ask ourselves, are there any blood cells there? How can you tell if any of those are actually blood cells? And remember, this is a video, so we're taking frames. Every 25th of a second, another frame comes in. If you have another frame and you have a black blob here in one frame and it's over here in another frame, is that the same cell? Is that the cell that's moved from there to there, or is it actually a cell that's moved from there to there? How can you track things when you can only see individual objects and we can't label each individual object? We can't say that's cell number 1, that's blood cell number 1, that's blood cell number 2, because all we know is that we've got a pattern in one frame and 25th of a second later, we've got another pattern of dots. Not only that, but remember, what I really wanted to do was to quantify how fast is the blood flowing through this capillary network. I wanted to be able to work out, for every vessel that I can see in the video, I want to know how fast the blood cells are moving through the capillary network, because that's what matters. But the capillary network itself is not necessarily static. We might have noticed that there's a little bit of movement in the previous image. But we're trying to work out how fast, or at least how well, the blood is perfusing the tissue and oxygenating the tissue. So we want to know how fast is a blood cell moving through a vessel that we cannot see. And the vessel that we cannot see is not necessarily static in the sense that from one frame to the next in the video, the vessel is not necessarily in the same place. So we've got an invisible vessel that might be moving as we take the video, and we're trying to work out what the blood cells are doing, which we can barely see at the best of times, what they're doing even when the video is unstable. And remember, we're talking about children, and if you're thinking about putting a microscope under an adult's tongue, you can just about get away with that. Try putting a microscope underneath the tongue of a 5-year-old and asking him to stay still. Then how is that microscope going to stay pointing at the same 1 by 1 millimeter of the capillary network? So in other words, before you do anything else, you've got to take into account the fact that the video is unstable. There might be lots of movement, translation, magnification changes, rotation. In other words, you might be actually looking at a different part of the microcirculation network, the capillary network. How can you guarantee that with a person's tongue, you hold that microscope and look at exactly the right point for a, ideally, 30-second period is what you would like. It would be good to have a 30-second run of video to see what these blood cells are doing. So it might be, if you're not an expert at holding this microscope absolutely still and telling the patient not to move their tongue, which instantly means the patient will move their tongue, because they'll say, okay, yeah. Basically, the image might be moving, it might be translating. But also it might be changing magnification, because we're looking at a 3-dimensional structure using a microscope, and if we push with a little more pressure on the tongue, which of course is not a fixed object. It will give slightly because it's tissue. If you push a little harder, you'll change the magnification of the capillary network that you're looking at. So the magnification might change as well. And how do you guarantee that you don't rotate the microscope? Maybe the person doing this experiment has got the microscope absolutely rigid, but then how do you know the tongue isn't changing ever so slightly as the video is progressing? And maybe there's distortion. Again, we're talking about a capillary network in a tongue. A tongue is a muscle, muscles flex. And therefore, regardless of any other changes, there might be distortions. The whole thing might stretch or squash or change. More likely, all of those are going on. And this person is sick. This person has got sepsis. So this person might be on a ventilator. And even if they're not on a ventilator, they will have a heartbeat. Otherwise, it's definitely too late to do this. So there'll be a heartbeat changing the way the blood flow moves. And basically, you have to try and take all of that into account such that you know what the capillary network is actually doing. And then, and only then, you can start to say, right, now let's see what the individual blood cells are doing and work out how fast they are traveling relative to the capillary network. So again, no maths, but it took me ages. It took me not weeks, it took me months to figure out how to process the video images in such a way that I could stabilize the network and then extract the information about how fast the blood cells are moving. I was given videos of people with sepsis, and I was giving videos of people who don't have sepsis as controls. In other words, healthy individuals. And I found that for a healthy individual, how many vessels in an image actually had a particular speed. These are the speeds in microns per second, so a big bump in the middle means that on average, blood flows through a human capillary in a sublingual microscope at about 0.5 millimeter per second. That appears to be what you would expect for a healthy individual. Some capillaries a little bit less, some capillaries a little bit more, but on average, 500 microns per second appears to be the speed at which blood flows through a capillary network. For a person with sepsis, you see it's rather different. It's not necessarily the fact that the average is a little bit lower, it's the fact that quite a few vessels do not have any flow. They have sort of zero flow, or flow that is so slow they are not very effective at oxygenating the tissue. This person has a problem. And the further this bump is over to the left-hand side, the more population of zero we have, the more severe the sepsis of this individual. I went back to the consultant and said, okay, I've analyzed your videos, and I think that for a healthy individual, it looks like my software is telling me that the average speed of a blood cell through a capillary network is about 500 microns per second. Is that right? And the consultant said, don't know. I'm not sure anybody's measured it before. All I know is that that video looks good, that video looks bad, this video means the patient is in trouble, this video means the patient is out of danger. Their expertise tells them, in other words, their expertise and their eyes and brain tell them, in this case, that looks like a problem. My experience tells me if it looks like that, they're probably okay. No one had actually quantified how fast blood cells move through capillaries. I found that staggering that no one had actually measured that before. So it is no surprise to find that sepsis is lower, but the fact that nobody had any numbers for these things, again, we're back to image analysis quantifying rather than telling you that looks good, that looks bad. Now we have real numbers. I didn't want to write software that produced histograms for anybody that was actually using this microscope. What I wanted to do was to actually produce color charts. In other words, anybody that actually did this, they have to be trained how to use the microscope, but ultimately I converted those numbers into, for each of these vessels I could see in this particular video, what was the average speed of the blood cells. Was it good, in other words, green to blue, or was it bad, red to orange? And for a healthy individual, it looks like the blood cells are moving quite nicely through the capillary network. We've got lots of greens, a few yellows, only one that looks a little bit reddish, a couple that looks a little bit reddish down here. And it's perfectly okay. Some individuals that are healthy will have some capillaries that don't perfuse oxygen too well. And the idea is that's what a control looks like, and if you have a person with sepsis, you get a lot more orange and a lot more red. So you don't have to be a rocket scientist to work out this one doesn't necessarily need any intervention. They're probably getting better. Whereas if they see this, they can tell the consultant, look, this guy's microcirculation, the circulation within the microcapillary network is getting worse. There's lots of red here. We need to ring the alarm bells and get this person on a different drug regimen or something like that. So I thought this might be useful for the NHS. Well, hopefully it will be useful for the NHS, but getting clinical trials is so difficult. So I wrote the software. It took me months to write the software, but arguably that was the easy bit. The next stage would have been, can we get clinical trials to run a consultant giving his or her opinion alongside the software giving its opinion as to whether or not this patient is getting better or worse. But the cost of setting up the clinical trial, we tried getting grant funding from various places, but the total cost, because it would involve a clinician, not a mere physicist who doesn't cost much, but a clinician would need to be involved in a hospital, the cost of getting the clinical trial set up would be somewhere of order £750,000. And we never got the funding to do this. So unfortunately, this I thought was a good idea, but it never made it into the NHS just because funding is such an issue. I haven't got long, I'm already at about an hour, so let me finish with the last bit, the bit that I'm doing now, which is cancer. And the group that I work with in the physics department at the University of Liverpool have been funded by the Engineering and Physical Sciences Research Council. We've been funded by Cancer Research UK, and most recently but funded by the National Institute of Health Research. And actually, we've got another Cancer Research UK grant, so maybe I could add that on the end as well. So we've had funding to do this sort of investigation. And what we're trying to do, basically, is ask ourselves, is it possible to get signatures of cancer based on infrared absorption? In other words, is it possible not only to diagnose cancer, you could argue that's the easy bit. If you've got a tumor, you've got cancer. The question is, can you look at tissue that hasn't yet become cancer and tell what's going to happen in the future? Prognosis rather than diagnosis. Why infrared absorption? Well, we had reasons to believe that if you shine infrared onto tissue samples, some infrared wavelengths will be absorbed, and maybe the particular wavelengths that get absorbed in healthy tissue are not the same as the wavelengths that get absorbed if you've got healthy tissue that will become cancerous. They may look identical now, but they are going to change in the future. Is it possible that infrared absorption could tell us that? The reason for infrared is because when you look at how molecules vibrate differently at different frequencies. Different molecules will vibrate because they're different masses of atoms bouncing backwards and forwards on different bonds. And infrared frequencies are a good match to the sort of frequencies at which molecules vibrate. That's why we think about infrared and don't think about what about shining visible light or ultraviolet light. Infrared happens to be a good match to the way molecules behave. So what we're effectively trying to do is to answer the question, that's the fancy way of saying it, but the simple way is, can we detect cancer before it's cancer? In other words, really early diagnosis. That's what we've been trying to do for the last few years. We started by thinking, where do we get the infrared light from? It started with going to Daresbury Laboratory, down the road in between Liverpool and Manchester. It's a laboratory with a number of different particle accelerators, and again, I'm not going to go through the detail, but if you wouse electrons around a ring here at close to the speed of light, they will emit lots of different radiation, including infrared. And if you actually get it to, if you get the electron beam to wiggle in a magnetic field, by getting the electron to wiggle backwards and forwards in a magnetic field, that will stimulate those electrons to emit electromagnetic radiation, which goes by the particular name of synchrotron radiation. So synchrotrons around the world are particle accelerators designed to produce electromagnetic radiation. We started by using one of these, bounced the lights off lots of mirrors, and started investigating the properties of cells that were grown for us by medics. As physicists, we rely on other people to provide us with the samples that we want to investigate. And we started looking at different cells and different tissues to start asking this question of, can we actually distinguish different tissues which ultimately have different prognosis? How do we know the prognosis? The medics provide us with samples, historical samples. They provide us with samples taken 5 years ago and tell us all of these samples are 5 years old and the patients are still doing fine. These are samples from 5 years ago, but the patients now have problems. In other words, we know the prognosis for different tissues which we have been supplied with. And we take infrared data, not simply imaging by taking an image of a particular tissue. This has been color-coded. It doesn't really matter what the colors stand for. But we can take an image at lots and lots and lots and lots of different infrared wavelengths. So in a sense, we've either got like a pack of cards, an image at this wavelength, an image at this wavelength, an image at this wavelength, or we can think of it as an image, but each pixel is not just a number, each pixel is an entire spectrum of what particular infrared wavelengths are absorbed or not absorbed by this particular tissue at this particular pixel. There's an enormous amount of information. Thousands of pixels, maybe millions of pixels in an image, and thousands of different wavelengths are scanned in what we call a data cube, because it's not simply an image, it's a whole stack of images. So there's a lot of data to be analyzed, and we're asking the question, can we pull out whether or not different tissues behave differently? I'm not going to spend time, because I'm already overrunning slightly, about what some of our PhD students have done. One of our PhD students in the last few years has done a wonderful job of showing that although optical images, of microscope images of tissues are very good at telling you the morphology, in other words, what it looks like, infrared tells you what particular molecules are present. And again, it doesn't matter what the blue and the yellow refer to. All it matters is that in principle, looking at a sample can tell you what it looks like, but infrared can tell you what it's made of. It's pretty much the same as with telescopes. You can take an image with the Hubble Space Telescope of a nebula and you can say, wow, that nebula looks fantastic. But if you take the spectrum of the nebula, then you can tell what the nebula is made of. And it's the same idea here. Morphology and appearance is one thing, but what really matters is what molecules are in this sample and how do they differ between this part of the sample and that part of the sample. And a PhD student did a wonderful job of fusing the information from both. The how does it look as well as what's actually going on. If those bits of information are fused together, we can get even more information. So what type of tissues are we studying at the moment? We're studying epithelial tissues. And by epithelial, that doesn't just mean your skin. It can mean, for instance, the esophagus or the mouth, where you have a mucous surface. We studied esophageal cancer for a while. We stopped studying it because we couldn't pronounce it properly. And then we moved on to oral cancer, and we're now working with OED, oral epithelial dysplasia. In other words, changes in the lining of the mouth. In particular, we want to improve the prognosis of if you see something in the mouth, what can we say about the likelihood of that progressing to cancer? At the moment, with all due respect to pathologists, if you take a sample which you think might be cancerous and you give it to a pathologist and ask them, is this a problem, yes or no, they don't get it right that often. Rather surprisingly, they get it right about 50% of the time. In other words, you may as well just toss a coin. The question is, can we do better? And the question that we really want to answer is this progression to cancer. We call it transforming or non-transforming. If you have what appears to be a lesion in your mouth, is it going to transform to cancer or not? In other words, is it going to be a problem or not? Here are two lesions in the mouth. They're the little white patches, the so-called leukoplakia. So there's a white patch, where's that? On the cheek on this person, and there looks to be a white patch on the tongue of this person. One of these is going to transform into cancer and going to be a problem. One of them is no problem whatsoever. But which is which? Coming back to the previous problem, you cannot tell by looking at them. One looks like, well, you know, that white patch looks a bit bigger than that white patch. No, that's not a good guide to what's going to happen. We are looking with the infrared spectra of lots and lots and lots of tissue samples that have been provided for us, generally with biopsies. It'd be nice to get rid of biopsies, which is one of the reasons we're doing this. We're being provided with biopsies and we're studying them to try and work out, is it possible to be able to tell just by taking a probe and touching it onto the white area here and getting a beep, yes, this is a problem, or a beep, no, this is not a problem. That's what we're ultimately working towards. But pathologists find it very difficult to tell the difference. And if we were to look at the infrared spectra, well, it's very difficult to tell the difference as well. So humans cannot look at an infrared spectrum of this or an infrared spectrum of that and say this one is going to be a problem, this one is not. But the differences are there in the spectra. We've got to find a way of extracting the information, not from the image, but from the spectrum, which is different for something that is healthy but will remain healthy and something that's healthy but will not remain healthy. How do we do that? Well, we do not want an army of PhD students to spend the next 30 years looking at those spectra, trying to work out what the differences are. So what do we use? Well, we use AI. AI has had bad press recently, but there is no way of doing this. There is so much data. We are talking about hundreds of thousands of spectra, each of which has got a lot of data points, and we're looking for tiny differences. Those differences are a reliable indicator, but they're small. And so we use artificial intelligence to try and extract that information. And because AI has such a bad name recently, I'll just spend the last couple of minutes just explaining what we're trying to do. If you want artificial intelligence to tell the difference between this tissue and that tissue, it's like basically asking artificial intelligence, can you tell the difference between a horse, an octopus, and a dog? Anybody in the audience would have no problem whatsoever in saying that's a horse, that's an octopus, and that's a dog. How do you know that's a horse? Can anybody describe to me how they know that's a horse? And the answer is usually, I just know. I've seen lots of them, and I know that's horsey. Horses tend to look a little bit like that. They've got 4 legs, a head and a tail. Okay, so have dogs. Yeah, okay. Horses don't have 8 legs, fine. But some images of octopus, you don't necessarily see all 8 legs. So you're quite right. How do you know it's a horse? Because you've seen lots of them in the past. So that's how artificial intelligence works. If you want to tell the difference of these 3, you show it lots and lots and lots and lots and lots of pictures of horses, as many as you can find. You show it different horses, different colors in different environments with different backgrounds. And if you show them enough pictures of horses and enough pictures of octopuses and enough pictures of dogs, hopefully the artificial intelligence will learn almost as well as a human does what those differences are. So then if you come back to it and say, right, I've trained it to understand the difference, I'm now going to validate and make sure that it does know the difference between those. I'm going to show it a picture it's never seen before, and it will say, based on all the pictures of octopuses I've seen in the past, I think that's an octopus. And basically, that's how it works. You validate by training what you think it wants to know, and then you check by giving it things and saying, what is that? And it comes back with a horse. You say, fine. And you say, what is that? And it comes back with an octopus. And you think, whoa, whoa. Why does it think that's an octopus? Well, that's part of the problem. Finding out why it's made the decision is critical. In other words, it's clearly got it wrong. Is that because the dog is running around in the sea? Is that because you can't count the legs on the dog? Is it because the tail appears to be coming out of its head? We can look at that and instantly say, doesn't matter. I can still see that's a 4-legged dog with a tail in the background and a head in the foreground, and it's in the froth of an ocean, and I can see that's a dog. Artificial intelligence doesn't necessarily understand the difference between an octopus, a horse, and a dog. It is simply based on the fact you've given it this data and it's making these decisions. But you can't ask it why. We are trying to apply artificial intelligence to looking at this tissue and looking at this tissue and deciding that this is going to become cancer and this isn't, and tell us why you've made that decision. And we are interrogating the output. I've had a PhD student working on this, and he is very good at writing AI routines and making sure that we understand them. They're not just black boxes where data goes in one end and something comes out the other. We will be interrogating it so that we can understand not only the fact that this is going to become cancerous, how confident are you that this is going to become cancerous, how confident are you this is going to become cancerous, and tell us how you've decided that that is what you think is going to happen. And that is currently what we're working on. So within the group in physics, we're doing this. We're developing an instrument which we're calling the Elder Wand. This is an instrument which ultimately, hopefully, a surgeon one day might be able to use this instrument, touch it on part of the patient or touch it on the inside of the mouth. Maybe a dentist could use this, touch it on the lesion, and it will come back in half a second and say, that's okay, no need to worry about that, come back in 5 years, or this is a problem, get it seen to immediately. So that's what we have been working on for the last few years. And by we, I remind you that science is never a solo effort. So we've had a lot of PhD students over the last 5 or 10 years, a number of colleagues in the department, and of course medics and other people. And thank you, of course, for the people who've paid for it, the funding bodies in the bottom right. Thank you all very much.